All right, today we're diving into a real molecular mystery. It's all about Huntington's disease and how scientists have just stumbled upon a brand new trail of clues. Clues that could totally change how we see this devastating condition. So, yeah, let's get right into it.
So here's the thing that's baffled scientists for decades. We know exactly what causes Huntington's disease. I mean, we've known it's a single faulty gene for a long time. So why can't we cure it? It feels like we should be able to, right? Well, the problem is, it's like we know who the culprit is, but we have no idea how they're committing the crime. We just don't get the step-by-step process that leads to brain cells dying off.
For more than 30 years, we've known who the main culprit is. The case file is crystal clear. A single mutated gene, the Huntington gene, or HTT, is the bad guy behind this whole devastating disease. Seems like an open and shut case, right? Well, not so fast. If we've had the culprit locked in our sights for all this time, why on earth do we still not have a cure? It's starting to look like knowing who the bad guy is isn't the whole story. Maybe, just maybe, we've been missing a whole layer of evidence that changes everything. Okay, so let's officially open the case file. What do we really know about Huntington's disease and this prime suspect that's so well known, yet somehow still so elusive? So the basics are pretty straightforward. This mutated HTT gene creates a toxic protein, and that protein basically poisons brain cells over time. This leads to a slow, progressive breakdown of a person's ability to move, think, and function. We know the cause, we see the tragic effect, but the part in the middle, the how, the play-by-play of the crime, that's where the trail goes cold. And that gap is exactly why a cure has been so hard to find. To find that missing evidence, scientists realized they needed a new kind of forensic tool, something that could look past the list of usual suspects and see what they were actually doing inside the cell. And that new tool is all about a process called phosphorylation. Think of it like this. Every protein in your cells is a tiny worker. Phosphorylation is the management system. It's how the cell sticks little instruction tags or post-it notes onto those workers. And these tags are basically just on-off switches, telling them what to do, where to go, and when to do it.
And this is a really important distinction. The old way, proteomics, is like getting an employee roster for a factory. You know who's there, but that's about it. This new approach, phosphoproteomics, is like getting the security camera footage for the whole factory. You can see who's actually working, who's on a coffee break, and, most importantly, who's got the wrong instructions and is about to mess everything up. It's a game changer.
So how did scientists even use this new tool on a brain disease? Now, to actually map these switches, the scientists did something that sounds like it's straight out of science fiction. They took simple skin cells from patients with Huntington's and reprogrammed them, turned them directly into neurons right there in a dish. Then, using this incredibly powerful technique called mass spectroscopy, they could see every single one of those on-off switches. And what's so important here is that these lab-grown neurons still have the patient's exact genetic code and even their age signature. So it's an incredibly realistic way to study the disease. And what did they find? Well, it was a huge breakthrough. The moment they looked at this new activity map, they saw a really clear, dramatic pattern of broken switches. Bingo! They found their first major clue. The entire communication system inside these cells was completely haywire. The first piece of hard evidence came as a number, 177. That's how many of those little cellular instruction tags were either missing or in the wrong place in the Huntington's neurons. This was the smoking gun, proof that the problem was way bigger than just one bad protein. It's so clear, right? The on-off switches are telling a much, much clearer story. It's like we've been trying to solve a crime by looking at a blurry, out-of-focus photo, and now someone just handed us a 4K, high-definition image. We can finally see what's actually going on.
Okay, so this heat map here, this is where we see the really specific evidence. Every little row is one of those molecular switches. Now look at the healthy cells on the left. See that big block of bright red? That means all those switches are flipped on, just like they're supposed to be. But then, look over at the Huntington cells on the right. That same block is solid blue. All those switches are completely, totally off.
And they counted them. There were exactly 27. 27 of these critical switches that were just broken in the Huntington's neurons. They were totally unphosphorylated, which just means they were stuck in the off position when they were supposed to be on. This wasn't a small tweak, you know? This was a total system shutdown for this group of proteins.
Now, in any good investigation, when you find a pattern, you look for the main culprit. And out of those 27 broken switches, one protein really stood out. It became the prime suspect in what we can call the curious case of MXR8. So let's meet our prime suspect, MXR8. The crazy thing is, before this study, nobody really knew what this protein did inside a neuron. It was a total unknown. And what made it so suspicious wasn't just that its switch was completely off. You can see the spot right there at serine 377, but that something else really weird was happening at the same time. And this brings us to a real paradox. Take a look at this. The switch for MXRA8, completely off, zero activity. And yet, the cell was making more of the protein. The total amount of MXRA8 protein was way higher in the Huntington's neurons. I mean, that just doesn't make any sense, right? The genetic instructions to make the protein were totally normal. In fact, the cell was actually making more of the MXRA8 protein than usual. But its on switch, that critical instruction tag, was completely 100% missing. The cell was working overtime to build a tool it couldn't even turn on. It's kind of like having a broken light switch in a dark room. The cell senses that something's wrong. The room is dark, so it panics and starts churning out more and more light bulbs, thinking that'll solve the problem. But it doesn't matter how many light bulbs you make, right? If the switch on the wall is working, none of them are ever going to work. So what happens to all those useless light bulbs? Well, they start to pile up. And that is exactly what you're seeing in these microscope images. In the Huntington's neurons, all that unused MXRA8 protein just starts clumping together. This isn't just a communication problem anymore. Now it's a physical problem, with junk piling up and clogging the cell's machinery. So the investigation is now zeroing in on this MXR8 protein. But was it acting alone, or was this just one piece of a much wider conspiracy? It was time to find its accomplices. To do that, they used a really neat technique that's basically a molecular magnet. They could go into the cell, grab onto all the MXRA8 protein, and pull it out, and anything else that was stuck to it would come out too. This let them see MXRA8's entire social circle. And it turns out, in the diseased neurons, MXRA starts hanging out with a totally different crowd. It starts binding way more to proteins that are part of the cell's waste disposal and recycling system. In the diseased neurons, MXRA was suddenly buddied up with proteins called TGM2 and LAMP1. Now, these aren't just random names. These guys are key workers in the cell's recycling plant. They're involved in taking the cell's trash bags and getting them to the incinerator, basically. So finding MXRA mixed up with them, that was a huge, huge clue. And that whole garbage disposal process has a name. It's called autophagy. It is absolutely essential for keeping a cell healthy. Now, here's the kicker.
Scientists have known for a long time that this exact system, autophagy, is broken in Huntington's disease. So the fact that our prime suspect, MXR8, is now physically linked to this system, well, that's a massive aha moment. It's the missing link. Okay, but there was one last piece of evidence they needed. This is the big one. All of this is super interesting in lab-grown cells, but does any of it actually happen in a real human brain with Huntington's? To find out, they had to go from the lab to the brain, looking at tissue from patients who had passed away from the disease.
And there it was, the final confirmation. Just like they saw in the dish, the neurons in the brains of Huntington's patients were clogged up with this MXR8 protein. You can see it right there, building up inside the outlines. This proved it. This wasn't some weird fluke in the lab. This is really happening inside the brains of people with the disease. So let's step back and think about why this is such a big deal. First, we now see a whole new way the disease works. It's not just about one bad protein, it's about a whole network of broken on-off switches. Second, we found a key link, MXRA, that connects the genetic cause directly to the cell's broken garbage disposal. What's also really interesting is that scientists are seeing similar broken switches in Alzheimer's, which suggests there might be a common problem across these terrible diseases. And maybe most importantly, this gives us new hope. These switches, they aren't just problems. They are brand new targets for developing future therapy. And that really leaves us with a huge question, doesn't it? For so long, the focus has been on trying to deal with that one faulty gene. But what if that's not the only way? What if the real key to unlocking treatments for Huntington's and maybe other neurodegenerative diseases is to figure out how to go in and fix these broken molecular switches? It's pretty amazing stuff. This new map of what's actually happening inside the cell has just blown open a brand new path for research. For the first time, we're getting a crystal clear look at the molecular crime scene inside a Huntington's neuron.